Our first speaker is Dr. Daphne Koller. Daphne Koller is CEO and founder of Insitro, a machine learning-enabled drug discovery company that she founded in 2018. Prior to Insitro, she was a computer science professor at Stanford, and she was also a trailblazer as the co-founder of Coursera, a pioneer in online education and courseware that I'm sure you all know about. Daphne's been recognized numerous times in her career. She was one of Time Magazine's, this is where I get to embarrass her. She was one of Time Magazine's 100 most influential people. She's a MacArthur Fellow, a member of the National Academy of Engineering, the National Academy of Sciences, a Fellow of the American Association for Artificial Intelligence, the American Academy of Arts and Sciences, the International Society of Computational Biology. And by the way, I know that she's also a leading force for AI at the World Economic Forum. Daphne is going to give a talk, and she'll be joined on stage after her talk by Zetta Managing Director Dylan Reed. Dylan for a fireside chat. Dylan leads our areas in AI native science, including our investments in AI native life sciences, and I think he's also standing behind me here. So Daphne, I'm going to hand this podium and this mic and clicker over to you.
Thank you.
Thank you.
That I wasn't expecting. Okay. Thank you, Mark, for the embarrassing introduction. It's a real pleasure to be here and tell you a little bit about something a little bit different than the traditional applications that one hears about for AI every day. We're going to tell you about how we can deploy AI not towards better chatbots, but towards better medicines and better health for people. So we know that AI is being built everywhere, and you can take existing applications and kind of put AI on top of them. But the question is, how do we build pharmaceutical drug R&D in a way that it is actually AI native? And I think that is the question that we set out to solve 7 years ago when we founded Insitro. So in order to deploy AI for that purpose, for deep science, deep tech, you can't just rely on data that is scraped from the web, because the data that we need to interrogate human biology and the drugs that truly modulate human disease, you need data that doesn't currently exist in most cases. You need data that is fit for purpose. And so fortunately, that too is a problem that we can today solve, because there is an increasing amount, as well as tools that are available to generate large amounts of high quality data from humans, from biological systems like cells, and from chemical compounds. And when we do that, when we put that data together and interrogate it through the lens of AI, we get novel insights into human disease that then pave the way towards better medicines. So, to highlight that transition point, I'd like to point to one of the most visible successes of AI in the field of biomedicine, of course, AlphaFold, Nobel Prize winner last year. And I'm not going to spend a lot of time talking about AlphaFold. I had nothing to do with it. But the success of AlphaFold really relied intrinsically on the existence of a resource called PDB, which is the Protein Data Bank, which is decades worth of accumulation of sequences and associated protein structures. And without that resource, a massive resource of data that had been available, this would not have been a feasible transformation. And so in order to make that happen, not just in the space of proteins, but more broadly in making medicines that modulate disease, we have taken a different approach that is very much an engineering-first approach, of which data is the foundational component. And we have built this incredible cellular data factory, you'll hear more about it later, that generates massive amounts of biological data across multiple modalities and multiple different cell types. That data is then fed into a software and compute architecture backed by close to 200 H100 GPUs, all of which interrogate the data through the lens of high-end AI and machine learning models. And then, of course, the people that make it all come to life, both on the computational side, but also life scientists who know what to do with all of these insights and how to turn them into actual medicines. So we use this technology stack to surface novel clinical and biological insights and turn them into new medicines for patients. Now, this is what's called the pharmaceutical R&D value chain. It's a very standard depiction. It starts from biology discovery, which gives us novel therapeutic insights, targets to go after, drug discovery, turn them into molecules, and at the end of the day, clinical delivery within clinical trials. In order to succeed in transforming pharmaceutical R&D, you can't just take a slice of this, because the entire value chain is currently really slow, really expensive, and has a dismal success rate of less than 10%. And that's only in the last chevron. That success rate is 10%, so it's really dismal. So I'm going to tell you how we've taken steps to transform every single one of those chevrons, starting with biology discovery, because if your program is going in the wrong direction after a wrong target, nothing you can do afterwards will possibly salvage it. You're kind of heading towards a brick wall and you just don't even know it. So in order to come up with novel, actionable, correct therapeutic hypotheses, we bring together two distinct data modalities, from humans and from the lab. So humans are experiments of nature, like all of us in this room, where we can take, at this point, really high content, high quality measurements from human anatomy, human pathophysiology, different types of imaging, different types of omics from blood and tissue, and clinical outcomes, and interrogate that richness of data using the lens of machine learning and AI. At the same time, we have the data factory that I alluded to earlier that prints massive amounts of data from living and fixed cells, and again, measures human biology as close as possible to the ground truth, unfiltered by the lens of a human thinking that they know what is important and what is not. And so together, we bring those two modalities to give us actionable targets that actually make a difference. Now, I mentioned the use of AI and machine learning in our platform. One fundamental place where this is used is simply see disease differently. Disease is currently defined using terms that humans knew to invent 30, 40 years ago from coarse clinical symptomology. Very little of that has to do with the underlying biology of the disease, which is why we still think of Alzheimer's disease as one disease, when undoubtedly there is so much more going on underneath. And so what we do is we apply machine learning models to the high content data that measure the disease at its very roots. Imaging modalities like you see here, omics modalities that are just harder to visualize, and create different axes across which the disease moves the patient from a healthy state to an unhealthy state, so we can then identify revertants that bring it back. So let me give an example of this. That is our lead program, which is liver fat and fibrosis, a disease that currently impacts over 25% of Americans, that many of them don't even know it, with the prevalence only rising with the increase of metabolic disease. So one of the challenges with liver disease is that it is underdiagnosed because people don't know they have it until their liver becomes cirrhotic, and by which point it's often too late to intervene. We decided to take the machine learning angle on high content data that is collected from individuals, MRI, which is the ground truth for this, but available only in very small quantities, as well as other modalities that no one would have thought could even tell you about liver disease because you can't even see the liver, like a bone scan, DEXA scan, and blood biomarkers and metabolites. And we were able to take the liver fat ground truth that we were getting from MRI and basically see it in a DEXA scan where any clinician could have said, you can barely see the liver. What do you mean you can see liver fat? Turns out that you can, and you can see it with very high accuracy. So what you see here are plots that show ground truth relative to predictions, and you can see accurate predictions across these three modalities, which then allows us to go back to these large human cohorts that I mentioned earlier and basically understand the genetic basis. And what you see here is a genetic association map that tells us which genes across your chromosomes have a strong effect on liver fat and fibrosis. And what you see here is that the more data you have, the more genetic insights you have, and that the machine learning, by allowing us to bring together these very rich modalities, has uncovered way more genetic insights at the bottom than were previously available at the top, which allowed us to identify a range of novel targets, 5 of which we took into animal models, and an incredible 4 of them actually validated in significantly reverting the disease in these mouse models. So this is one example. Very quickly, I'll show you another one. So this is the platform that we've built for cellular data generation. And I will show you that in the context of a devastating disease, which is ALS, also called Lou Gehrig's disease. But first, let me show the platform. So you can see this is what an automated lab looks like. You can see the robot performing the experiments, moving the plates, perturbing the plates. And at this point, we can take an entire experiment from a stem cell, differentiating into, say, a neuron, perturbing it using CRISPR, measuring it using imaging, measuring it using omics. And the entire process, including data pipeline, is not touched by human hands. And this is sort of a graph that shows the amount of data that's been accumulating in our lab from the time of inception. Early days is when we built the platform. Things were slow, but now we're seeing a 12% growth month on month in the amount of cellular data that the lab produces. So here's the work that we did in ALS to show you a slightly different discovery approach. What you see here is the disease axis that I mentioned earlier, in this case in a cellular surrogate for ALS, a motor neuron that is created in a dish. You can't really measure the disease inside people's brains. People don't like it when you poke needles into their brain. I understand that. And so we made those neurons sick by perturbing them in the same way that the disease does. And what you see here is an image that shows how the neurons change when they go from healthy to sick. And you can visibly see the changes in this machine learned disease axis. Which allowed us to then screen for revertance of that machine learned disease axis. And what I'm showing you is the results from our very first target that is now very rapidly making its way towards the clinic. And you can see that in a completely separate follow-on experiment, it reverted the neuronal outgrowth deficit that you see in ALS. So on the left side, healthy neurons, you cut them off, they grow back. In the middle is the disease state. You cut the neurons, they don't grow back. And on the right side, with our intervention, you cut the neurons in the disease state and they do grow back. And this is the first time that we've seen this type of functional reversion of a disease phenotype like this. And we're super excited about this program. So great targets come out in fatty liver and fibrosis. They come out in ALS, other programs that we have as well. What do you do with them? Now you need to actually make molecules. And that, too, is a really hard problem. So let me tell you a little bit about the platform that we've built for doing chemistry, taking targets and turning them into molecules. And what you see here is all comes down to the data, and there's just not as much data on small molecules that bind to proteins as there is for proteins that fold. And so we had to build our own data engine. And so what we did is we constructed, using a technology called DNA-encoded libraries that allow you to interrogate billions of molecules at the same time in a single pooled format in terms of their binding affinity to your protein target. And so you are basically doing 1 billion experiments at once in a single test tube. That gives us one set of models that you care about. The other set of models is what's called ADMET models or PK models, which are the pharmacological properties that turn a binder to a drug. Because at this point, there's a bunch of different technologies that get you a good binder. Some of them experimental, some of them computational or a hybrid. But then there's like 3 years of work to take that small molecule and optimize all of the properties that make it go into the body, get absorbed by your gut, circulate appropriately in your bloodstream, not get secreted by your kidneys. All of those are years of optimization. And so we've built a set of models for that as well. We have an active learning, machine learning engine that tells us once you have these models, which compounds to synthesize, which ones to test, and then learns quickly from the result of that active learning lab in the loop in order to synthesize the minimum number of compounds that are necessary in order to get us to the molecule that actually works. This is an illustration of the kind of machine learning that you can do when you have this kind of active learning kind of lab in the loop. What you see on the left is a model that was trained on just this naive DNA-encoded library data and asking to what extent can we predict for a new molecule whether it's actually going to bind to the protein. And it's honestly a little bit random in the performance. You take that same model and you feed it the data from this second generation library, which is informed by the model and the first generation set of results. And all of a sudden, in a single experiment, you've developed an entire model that tells you exactly well, not exactly, but with fairly high accuracy, which molecule that you have not yet synthesized is going to actually bind to your protein target and with what binding affinity. So you can now do most of your design in silico without the long and laborious process of synthesizing thousands of small molecules and testing them one at a time. So in order to further enable this, we are excited to have announced yesterday, so this talk is very timely, a wonderful partnership with Lilly, which has provided us with one of the industry's most valuable data sets used to train an AI system. It is decades worth of chemistry data that measures exactly those pharmacological properties of solubility and absorbency, and does it get into cells, and all those other things that are so hard to come by if you're a small biotech company. So we're working with Lilly to create machine learning models that will serve not only us and Lilly, but also their partners and our partners in order to predict those pharmacological properties and hugely compress the time that it takes to get from a binder to a drug. And more importantly, even than time, it's the ability to avoid going down the rabbit hole that's going in the wrong direction to something that will never become a drug, and then eventually saying, you know what, I'm just gonna have to compromise because I'm not going to get within the time constraints of my project a drug that works, so I need to compromise on a suboptimal drug. We can short circuit all of that. And so those properties, those molecular property models are not only faster and cheaper, but more importantly, better. And then finally, once you do that, once you have a drug, you need to take it into the clinic. And we've done a ton of work on predicting subtypes of individuals and early onset of disease so that we can maximally power our clinical trials and intervene in disease when it still makes a difference. And so what you see here, for example, is the ability to predict amyloid beta in the brain as measured by a PET scan, but without doing the PET scan, just from a standard of care MRI so that you can diagnose people very early in the disease from standard of care data. This is the same thing for AMD, which is an eye disease, and you can see that our predictor has a much higher correlation, for example, with visual acuity than an actual clinician diagnosis, and again, allows us to diagnose this disease far earlier than a clinician can. So all in all, AI will transform every step of the value chain. We've already made significant headway in every single one of those and are continuing to improve and optimize our engines. It takes a village. We have not done this entirely by ourselves. We have wonderful partners on the biology discovery, specifically some of those national data banks, partners like Lilly on the chemistry side, BMS, which has been a wonderful partner in our ALS project. And this is the drugs, because at the end of the day, it all comes down to patients, and what you need to serve patients is drugs that will actually help them. And so we have a really exciting pipeline across multiple indications and are excited to take our lead program into the clinic next year. All in service of our mission, which is to bring better drugs faster to the patients who can benefit most through machine learning and data at scale, or as we like to say, pipeline through platform. Thank you very much.